Hi, I'm Catherine White, an assistant professor in the Department of Chemistry and Biochemistry at the University of Notre Dame. I'm excited to talk to you about the work that my lab has been doing for the last few years on developing new tools to raise pH in living cells and study pH-dependent cell polarization and migration. Intracellular pH or pHi dynamics regulate normal cell behaviors. Transient changes in pHi on the order of 7.0 to 7.6 have been shown to be necessary or sufficient for diverse cell processes, including cell adhesion and migration, proliferation and differentiation, and cytoskeleton remodeling. However, many of the data supporting the role of pHi in these processes have been collected under non-physiological culture conditions and using non-specific pHi manipulation techniques. Thus, we need better tools to understand the role of pHi in regulating cell biology. Our lab investigates roles for spatial and temporal pH dynamics in regulating cell biology. One example of a behavior where spatial pHi dynamics may be important is in a wound healing assay. Here's a normal wound healing assay conducted by Caitlin Donahue, a graduate student in the lab. As you can see, cells migrate rapidly into the wound edge, and you can see leading cell phenotype and follower cell phenotype. If we pin pHi low, migration into the wound edge is inhibited. However, it's not known how spatial pHi dynamics at the leading edge versus in follower cells may contribute to this effect. So this leads to the question of whether high pHi drives the leader cell phenotype. In order to answer this, we need a tool that allows single cell spatial control of pH, long-term manipulation on the order of hours to days, and one that ideally is non-damaging and non-toxic. We also study temporal pHi dynamics in processes like cell cycle progression. This is data collected by Julia Spear, a graduate student in my lab who had a talk here earlier at ASCB. Julia's imaging using a pH biosensor pH dynamics during cell cycle progression, and she can back calculate the absolute pHi during this process of cell division. And you can see cells undergo prophase, cytokinesis, and the resulting daughter cells. pH is dynamic during this process and oscillates temporally during this process of cell cycle progression. So this leads to the question of, does dynamic pHi time the cell cycle? And in order to test this mechanistically, we need a pHi manipulation tool with single cell temporal control and one that's ideally reversible to mimic these oscillations. Now, I won't go into too much detail, but there's no existing tool that meets all of these criteria. So for the last several years, we've been working on developing new tools for spatial and temporal pH manipulation in single cells. Caitlin Donahue has adapted a tool developed as a neuronal silencer by Ed Boyden's lab. It's called ArchT, and it pumps protons across the plasma membrane when stimulated with 561 nanometer light. And it has previously been used on very short timescales, but Caitlin has adapted this for longer timescales to raise pH in single cells. And in all of the data that I'm showing, we use genetically encoded or dye-based pH biosensors to read out intracellular pH. So the first thing that Caitlin did was test whether the expression of the tool alone altered pHi. Luckily, she saw that there was no difference in pH in the absence of stimulation between the ArchT expressing cells and the control epithelial cells. So then what we did is use a confocal microscopy approach to measure the pH biosensor across a whole field of view, and then used localized photomanipulation to spatially activate ArchT only in the red box. And so for the rest of my talk, if you see a red box, that indicates the spatial region that we're using the activating wavelength of light. And so as we start illumination, you can see the fluorescent intensity increases, indicating an increase in pH, but only in the stimulated cell. And we can track this over time and use image analysis approaches to quantify that pH increase as read out by the change in fluorescence of that pH biosensor. So only in the stimulation ROI and the resulting stimulated cell, cell 2, do we see an increase in that biosensor fluorescence. The unstimulated cells are unaffected. So Caitlin collected data over many, many cells and found this to be highly reproducible and consistent. Increased pHi required both ArchT expression and light stimulation. And under these conditions, ArchT can raise pH by about 0.18 pH units. But Caitlin showed in her paper that this was titratable with light intensity. And so using various LED lights led to increases between 0.1 and 0.4 pH units. Finally, she showed that repeated stimulations can cause PHI increases of up to an hour. So we can study biology on the hour time scale. So the first proof of principle Caitlin did was test whether single cell PHI increases could drive single cell membrane protrusion events. One hypothesis is that protons are diffusible, so local activation of ArchT, which raises PHI across the whole cell, would produce global membrane protrusions. However, it's also possible that local protein recruitment in response to PHI increases could produce a localized membrane protrusion event in response to that pH change. So for the first time, we have a tool in hand that can actually address this question. So here I'm showing cells that Caitlin stimulated locally in the red circle, ArchT on the left and control cells on the right. And what we noticed immediately is that PHI increases with ArchT drive increased membrane dynamics compared to the control. And surprisingly, those dynamics are local with significant protrusion events just around and within that stimulation ROI, whereas the control cells are unaffected. Now, Caitlin characterized and quantified this in her paper, so I encourage you to check it out if you're interested. But now what she's doing is investigating whether we can simultaneously monitor actin dynamics and pH-sensitive protein recruitment during these pH-driven membrane protrusions. And so what we did was stably express LifeAct GFP to label actin in the cell. And again, we can see this membrane ruffling and protrusion event that occurs in response to light stimulation, whereas the rest of the cell is again relatively unaffected. But now we can actually track those actin dynamics during extension and protrusion and retraction in the stimulation ROI and compare that to the chymograph from LifeAct in the unstimulated region of the cell at the rear of the cell. Again, PHI increases across the whole cell, but actin dynamics are local. Microtubule dynamics are not pH sensitive. I didn't have time to show that data today, but we've demonstrated that on this time scale. And Caitlin's able to see new rear end retraction in about 30% of cells. This means that when she induces a protrusion event in about 30% of cells, she sees eventual retraction of the rear of the cell, indicating a role for pH in changing cell polarization. And so the next question that she's addressing is whether pH-sensitive actin cytoskeleton remodeling proteins are locally recruited with this stimulation. So to sum up this section, ArchT can raise PHI by 0.1 to 0.4 pH units and for up to 1 hour. It can drive localized membrane protrusions and dynamics. The limitations of archirhodopsin are that it's electrogenic, which means it also alters membrane potential as well as intracellular pH. So that is a significant concern with our study of these biological behaviors. And another issue is that ArchT requires near constant illumination. And although we use low power LED, there's potential issues with long-term illumination of your biological samples. So we next asked whether we could modify a ubiquitous electroneutral ion transporter and apply it to spatially temporally raise PHI. So this second generation tool relied on the engineering of the sodium proton exchanger, NHE1. It's a key regulator of intracellular pH homeostasis and is activated by low or acidic PHI that then leads to the co-transport of an intracellular proton in exchange with an extracellular sodium. As a postdoc, I found with collaborator Bradley Webb that histidines in the C-terminal tail regulate this activity. And at low pH, they're protonated. The tail is recruited to the inner leaflet of the plasma membrane where it binds to negatively charged pips. As the activity re-equilibrates PHI, those histidines become deprotonated, the C-terminal tail disassociates from the plasma membrane, and this silences NHE1 transport activity. So I proposed using a light activatable module in combination with this ion transporter to control activity. This utilizes LOVE2, which is a light activatable domain, and ZDK, which is a peptide that binds LOVE2 only in the inactive, no dark conformation. Upon stimulation with light, LOVE2 undergoes an unfolding, ZDK no longer binds. And so we proposed using this to control association of the C-terminal tail with the inner leaflet of the plasma membrane. Using light activatable modules with the addition of light, we can induce dissociation of the LOVE2 and ZDK and allow association of the C-terminal tail with the inner leaflet of the plasma membrane to turn on enzyme activity, transporter activity. And so Keelan Trull, a postdoc in my lab, has been working on this project. We were finally able to see good expression of the optogenetic tool in these otherwise NHE1 null cells. So Keelan then tested whether opto-NHE1 cells exposed to light could have an increase in PHI. So using fluorescence of the pH-sensitive dye, we saw an increase in this dye fluorescence, indicating an increase in pH when we had opto-NHE1 cells that were exposed to light. When those opto-NHE1 cells were not exposed to light, there was no change in pH over this time course. And importantly, control cells that were stimulated with light similarly had no response in terms of their PHI. So what this shows is that PHI increases require both N-opto-NHE1 expression and light activation. So what I've shown you today is that the N-opto-NHE1 has light activatable activity and can raise PHI in single cells by up to 0.6 pH units. And this induces an electroneutral increase in PHI, which is a distinct advantage over the ArchT. Now, some continued limitations of this tool is that the robustness of the response is highly dependent on the surface expression. And this tool still requires near constant light activation to maintain that high PHI. Keelan is doing ongoing protein engineering to further improve and address these two limitations. First off, he's identified a library selection, which allows us to enrich NHE1 library pools for active NHE1 using an ammonium chloride load. With increasing rounds of selection, you can see we're enriching for NHE1 expressing cells over our entire cell population. He's also able to select against NHE1 activity using an approach of lithium chloride poisoning to select for cells with inactive NHE1. So you can see here the population of NHE1 cells decreases over these selection times. So Keelan is applying this approach to screen a library of split NHE1 variants where we can use light to reconstitute activity. So I mentioned the people who worked on this project throughout the talk. I'd also like to mention funding sources, including an NIH New Innovator Award and these internal sources of funding for graduate students and myself. I'm also recruiting postdocs, so please send a CV and cover letter if you are interested. I thank you for your attention.